Let us proceed with chapter 2, that is the sexual reproduction in flowering plant. For that we have to study the flowers, which are the parts of the plant treated as sexual part, sexual organ. Actually, what is flower? आप जानते होंगे फ्लावर क्या है. अक्सर जो डेफिनेशन दी गई है वो ये है, फ्लावर इज द reproductive branch of a plant. Now question is, the shoot, the branch, it consists of a stem and leaf. Now in flower, leaves are modified into sepals, into petals, into androecium, into gynoecium. Stamen is the male part and that develop from microsporophyll, and pistil is the female part that develop from megasporophyll. In this way, flower is a modified shoot which develops from a special apical meristeme. For developing a flower, a number of hormonal, mesmetic, embryological, structural transformation occur prior to initiation of flowering. Shoot apical meristeme is transferred into reproductive meristeme. It grows to form inflorescence axis over which flower primaria develop and they grow into floral bud and that mature into flower. Let us study development of pollen sac. Pollen sac, they are present in anther. This is the anther. And this one is the filament. This anther has two lobes and each lobe contains two pollen sacs. In this way, each anther is tetrasporangiate. This is the two lobe of anther. This is the one lobe and this is the another lobe. This lobe has two pollen sacs. In this way, this anther has four pollen sacs, four microsporangium. These are four microsporangium. That's why it is called tetrasporangiate. Sporangiate. If you see a transverse section of young anther, then this young anther lobe this is the young anther lobe. This outermost covering This is called epidermis. Epidermis. Then hypodermal cell at each corner. These hypodermal cells get enlarged and these are called archisporium. Archisporial cell. This archisporial cell get split into two. Outer is called parietal cell and inner is called sporogenous cell. Outer, this parietal cell, it undergoes pericanal and anticanal division, and then this is parietal cell, and this one is the sporogenous cell. Parietal cell and sporogenous cell. Here is the epidermis, and then this parietal cell, it develops wall of spore sac. If you will see wall, then there is the outer wall. This outer wall is fibrous in nature. This outer wall is fibrous in nature. This is called endothecium. This at certain position, this endothecium is thin-walled. At this position, stomium develop. This stomium is a position from which this pollen sac get burst, get ruptured, and pollen grains are liberated, and it is followed by middle layer, it consists of 2 to 3 layer, middle layer, and cells are thin-walled. Cell, they get crushed, and then the innermost layer, that is tapetum, it is radially elongated cell. Tapetum. These cells are radially elongated. They may be multinucleated, have a large nucleus. They may be polyploid. They provide nourishment to microspore mother cell. These are the microspore mother cell. Microspore mother cell. This tapetum has many functions. This tapetum provide Ubis granule. These Ubis granules, they provide sporopollenin. the hardest material that form engine of pollen grain. Engine of pollen grain, and this tapetum it secrete IAA, that is the auxin, a plant hormone. This is for multiplication of cell, especially microspore mother cell. This tapetum provide nourishment to microspore mother cell. Microspore mother cell, it provide nourishment. It provide a sticky material which is fatty, known as pollen kit. These pollen kits, they are sticky in nature. Due to presence of this pollen kit over pollen grain get adhered at the surface of insects, and they help in pollination. Pollination by insect is due to this pollen kit, and in those cases where pollination is not done by insect, in that case pollen kit is absent around the pollen grain. As for case anemophilous plant, the flowers which undergoes pollination with the help of wind, they are comes under category anemophilous, and pollen kit is absent. Hydrophily plants in which pollination is done by water, this pollen kit is also absent. And ultimately, if you see the structure of a mature anther, then you will see this is the stomium. Stomium develops from outermost covering, that is the endothecium, which has fibrous layer, but few cells are parenchymatous thin wall that provide site for stomium, at which place this pollen sac rupture and spores are liberated. Middle layer somewhat get crushed. The tapetum, the innermost layer, get consumed, that provide nourishment to microspore mother cell. Microspore mother cell undergoes reduction division to form microspore tetrads, and each tetrads later on get developed into microspore. Microspore is the pollen grain that has single nucleus. Later on it undergoes division, asymmetrical division, unequal division, that results into two cell, generative cell and vegetative cell. This is the two cell stage, and in most of the cases pollen grains are released at this stage.